for the invitation. It's definitely a fun little theme we got going on today because we definitely have a lot of similarities in common. But I think, in terms of the how skin damage can kind of lead to these kind of systemic effect throughout the body, and it's not just localized to the skin itself. Um, but whereas Miriam was kind of looking more on the innate side of things, and how that could influence kind of more of the innate inflammation with the bone marrow effects, we were kind of more interested in more of the food allergy side of the kind of triad, and how it can actually change the way we think about sensitization and the adaptive immune response. And so traditionally, Andrew's lab, where I did my PhD, basically he likes to challenge a lot of the assumptions and really understand some of the foundational aspects of how the immune system works. And so to kind of go through those lines, one of the foundational principles of the adaptive immune response is this kind of three signal hypothesis, where signal one is the target itself, the peptide MHC. The second signal is basically the licensing signal, is how do we know when we should even mount an immune response. And only when we have both of those signals together, license and a target, do we actually induce an adaptive response. And then the signal three is the cytokines that direct what kind of immune response. And so a lot of work has been, really focused on what is signal two here, because, that is what is really controlling whether or not we have an immune response, which, is, we want it to happen, against bacterial infections and things like that. But in the case of autoimmunity, it's, something we don't necessarily want to have happen, and needs to be tightly controlled. And this kind of investigation into what these licensing signals are has been particularly fruitful for a lot of infectious agents, small microbes, mostly, bacteria, viruses, some fungi as well. And what this kind of led to is the identification of these, predefined molecular patterns that have been evolutionarily just kind of kept similar, and using, because of their kind of evolutionary conserved features that are kind of attached to the microbes themselves. The immune system developed these pattern recognition receptors such as TLRs and LRs, and the like, to basically identify these conserved moieties that are physically attached to the microbes themselves. So that way, when a phagocyte kind of consumes the microbe, it has both the kind of the signal to be able to license it, as well as the signal one, which is all the protein targets attached to the microbe themselves. But what about a type 2 response? And when we think about what actually induces a type 2 response, you can see that it really kind of covers a different class and scales of biology here, or even chemistry, in the sense of toxins can cause it, proteases, unicellular organisms, and multicellular organisms. So when you have all these different kinds of classes of things that can induce a type 2 response, it's really hard to find something that is evolutionarily conserved. And so a lot of the same techniques or thinking, in terms of trying to find specific receptors, has been a little bit more difficult. And some people have tried to find specific receptors to try to kind of mimic the same way we thought about, antiviral responses. But it doesn't really pan out as well, principally because of the fact that there's not a lot of similarity in terms of a lot of the features that can cause these responses. So instead, we thought, all right, if there's not a specific moiety, kind of molecular feature on the kind of insult itself, is there something else that unites all of these things? And principally, what we thought of is just that they all cause some sort of damage, principally to the epithelium, but also, if they were to get inside, to the parenchyma itself. And so we basically, have to think about, all right, how can we relay the information that there's damage, um, and do different types of damages, kind of relay different types of information to skew responses in one way or another. And then, if there is some sort of host interpretation of a damage relay, how can we make sure that it's targeted towards the actual target that is causing that insult? And so we kind of put these questions into the context of food allergy, and kind of Erin kind of went over this already, but basically the idea, right, is that if there's some sort of mucosal damage, you kind of get this release of these alarmins, IL-25, 33, TSLP, also sometimes IL-1. And at the same time, you get this translocation of food allergens. And the idea is, again, also here it's kind of it's depicted frequently that these alarmins are just kind of spewing out from the epithelium, but they're actually localized in very different regions. And so it's kind of be interesting to look at how exactly this all happens. But the idea is that the phagocyte that will eventually be the presentation cell will interpret the kind of alarmin signal for licensing and also pick up the allergen that comes across. And then it'll go to the draining lymph node, prime TFHs and TH2s. And then along with other innate cells, we don't really know exactly who is making everything that's important here, but provide the signal 3 to induce the Ig switching that gets loaded on mast cells that cause a lot of the food allergy reactions. And so while this was kind of originally depicted as happening in the gut, what people think about now is that the skin is actually a major source of allergic sensitization. And that's primarily due to the fact that there's a strong associations with atopic dermatitis or use of kind of lotions or creams on the skin. Um, and so a lot of that epidemiology kind of pointed that direction, and people were able to take that and put it into mouse models, where you can either cause skin damage by tape stripping the skin or some other kind of related models, and add it to allergen on top of that damaged or challenged skin, and basically you get a local response in the skin itself, which we kind of start to think about it as a local priming effect in the skin. Um, but when we think about, you know, how exactly these pieces are coming together from a more molecular cellular perspective, one of the differences between this antigen presentation cell that is different from a microbial infection is that the kind of trigger that licensed the APC is not physically attached to the allergen or the molecular target. So signal 1 and signal 2 are actually physically unlinked. And so what I was kind of trying to get to address before is that this APC has to be able to find both of those. And it's not clear whether those things are coming in the exact same space or how close they're coming. And then, right, kind of as Miriam was suggesting, that a lot of skin damage can actually, even though it's caused locally in the skin, can end up having systemic effects, or at least go into the serum. Um, and so, if that's true, and these alarmins are getting into the serum, what can they cause, in other parts of the body, specifically more as it relates to an adaptive response? Because a lot of people, have been looking at how it affects the effects of systemic inflammation on cell activity, but never really focused clearly on whether it can actually induce an adaptive response itself. And so kind of as, another example, um, of data that basically kind of led us along this thinking of, all right, yes, there is alarmins coming into the skin after acute injury. And also when you look at patients with atopic dermatitis, you can also see alarmins, TSLP and IL-33, being elevated in the serum, right? So basically, we wanted to ask, all right, if these are getting to the serum, and they might have the power to kind of induce adaptive responses, can there be a situation in which um, there is skin damage that causes the release of a lot of these cytokines that waft over to a different organ system and actually prime a response to an antigen that had nothing to do with the original insult in a very different space. And this is kind of what we would kind of consider a little bit more of a remote priming situation. And so we decided to test this directly, kind of in mice, where we basically adapted the tape stripping model of allergic sensitization a little bit differently, where we did the tape stripping on the back, and definitely a lot more aggressively than Mariam did, to cause a robust amount of skin damage. But instead of putting the ova on the damaged skin, we only introduced it by gavage. And we did this 3 times and then, took blood for looking for antibodies and did an IV challenge to see if those antibodies were actually productive. And when we did this, we actually did find that the tape stripping on the skin could cause this anaphylactic-like response when the mice were then challenged with ova. And importantly, when we did this in an IL-4 deficient mouse, this anaphylaxis goes away, suggesting that this is an allergic kind of feature and not some other random thing that's happening. But of course, the tape stripping is kind of one random thing that we use a lot and may or may not have a lot of intrinsic value as it translates to humans and things like that. So we decided to try a different kind of set of modalities for causing skin damage to see if the same thing was true. And so we decided to test a few things, primarily an MC903 calcipotriol-driven dermatitis, where this, the dermatitis was given a little bit more chronically, and we kind of fed the ova throughout, was also able to cause this. We also kind of came across basically injecting acetone intradermally, which caused these little blisters on the surface of the skin, which again was a very robust response. And then as well as a hole punch through all the layers of the skin. And then finally, we tested a UV burn to kind of mimic sunburns with the idea that basically that UV was technically, I don't know, and at least in the literature, more kind of associated with type 1 inflammation, a lot of interferons and things like that. So we thought maybe this might skew away from any kind of allergy response. But in fact, it was a very similar compared to the rest of the other types of damages that we had done. Um, and of course, up to this point, we're kind of looking at anaphylaxis. But when we take antibodies and basically try to, look at how much they go compared to just the ova alone controls, we do see that each of these types of skin damage can remotely prime ova that has been introduced in the gut, and generally tend to produce allergy-associated antibodies, IgG1 and IgE. And clearly, you can see that each kind of skin damage has a slightly different pattern of intensity between the allergies. But generally speaking, they all produce enough, right, to induce that anaphylaxis response. So right then, how is it if we can see the antibodies, we can see the anaphylaxis, but how is this signal actually being transmitted? And because of the way we kind of started to think about things, we first thought to basically examine, whether, there's cytokines, inflammation that's passing intravascularly to induce this effect. And we did this with a two-pronged approach. The first is to see, which cytokines can even do this when they're present in the blood. And then secondly, which cytokines are even there when we cause the skin damage. And so we basically tested this first half by injecting the mice intravascularly with a cytokine of choice, and then giving ova in the footpad. Again, a different kind of remote situation that was just easier to screen with. And basically, we did a lot of different cytokines that are kind of, classic immunology favorites. And what we found was basically that it was mainly the kind of IL-1 family members, as well as TSLP, but interestingly, not IL-25. That kind of completes that alarmin class. They were able to make IgG1 and IgE. And it was kind of also cool that the IL-1 and IL-18 were more capable of producing a lot of the other kind of antimicrobial associated antibodies, whereas the TSLP and IL-33 were a lot more restricted to the type 2 associated antibodies. And so there's this kind of interesting, specialization of some cytokines to be more allergy focused, whereas 1 and 18 are kind of more pan focused or capable, or not unfocused really, and making every kind of antibody. And so clearly, these four cytokines are capable of inducing IgE, but what are they even there? And so we basically tried to do a multiplex cytokine analysis in the blood 1 hour after these different types of damage on either the first time we sensitized or the second. And basically what had popped up again, there's these four cytokines are there, at least one, if not multiple of them in each of these different types of damage. And of course, all the other cytokines are also kind of presenting themselves in different patterns, which we believe is kind of influencing the different forms or variations in the antibody isotypes that are produced. And right, so it seems like the cytokines are sufficient to cause IgE, and they're definitely present in the skin or in the blood after skin damage. But are they actually important? And so to do this, we basically tested these models in mice that were deficient in these pathways. So here the TKO being a knockout for IL-25, ST2, which is the IL-33 receptor, and the TSLP receptor. And when we do this here is with tape stripping in these triple knockout mice, we lose all of the effect. And this is true for all the other models that we kind of utilized in the study. And sometimes, we only need to knock out one or two of these to actually have it happen, or to have it, be lost. And so, to kind of summarize this aspect of things, we do think that at least one mechanism that this could be happening is that, this skin damage causes the release of, these cytokine mediators into the blood. And predominantly IL-33, IL-18, IL-1, and TSLP have this IgE inducing capacity. And basically, we think this will travel to kind of an external lymphoid organ, which will then kind of join up with the antigen signal 1 coming in to induce an antigen-specific IgG1 response. And of course, because of the fact that tape stripping or any of these damage models has a lot of these other cytokines present, we wanted to be able to try to focus on how can we understand this better by just focusing on one kind of signal itself. And so we chose IL-33 mostly because it was very potent at inducing the IgE response. And so basically did the same kind of setup as before, where we inject IL-33, but this time we're putting ova by gavage into the stomach. And we did this once, twice, or three times, and we could see, similar to with the footpad, the induction of IgG1 and IgE in an antigen-specific manner. But it tended to take at least two different sensitization periods to actually get the ova-specific IgE to show up. And what's kind of neat about this kind of model, as opposed to some other allergy-inducing models like alum, is that it's really flexible. So we can put IL-33 IV, put the ova intranasally in the footpad in the stomach, and basically get sensitization to happen. You can really modulate based off of how much ova you put in, how much IL-33 you put in, can really titrate how much antigen-specific IgE you get out. And what's also kind of interesting is that if you inject IL-33, you can wait somewhere between 6 and 8 hours to inject, or to introduce the ova, and still get sensitization to happen. So it definitely doesn't have to be a coincident event, but rather IL-33 can induce a priming in these APCs that will last for a couple of hours in order to kind of grab their target and present it. And of course, when I say that, people tend to get scared in the sense of like, all right, any skin damage is going to sensitize me to anything, a few hours later. But again, this has all been models where ova is introduced as a novel antigen. Most of the time, we're already eating food that we've eaten before. And so we decided to test this directly of whether IL-33 can break tolerance. And so we did had mice on either water or ova in the drinking water for a couple of weeks, and then we sensitized. And basically, when we had the tolerization group that drank ova beforehand, we could not get any sensitization to happen, either through IgE or anaphylaxis, suggesting that IL-33 is not breaking tolerance, just kind of being able to license it the first time you see the antigen. And so, we clearly see that something's happening. But now the question really is, who's kind of starting this cascade, or how is this even happening at a cellular level? There's a lot of different cells that can express the receptor for ST2. And so we kind of wanted to figure out who was the main driver of this. And so we basically just took the conditional or knockout approach with, the wild type or the floxes, as a control, and then the full knockout where you don't get any response whatsoever. And basically, we did not see any effect if we had an eosinophil deficient mouse, a mast cell and basophil depleted mouse, Cas-based knockout, a CD4, T cell depletion or deletion of ST2. Also didn't do anything. And what was interesting is that when we tried two different pre-drivers to delete in dendritic cells, which are primarily thought to be the main antigen presentation cell, and this had no effect, which is very different than other alarmins or other kind of inducing cytokines like TSLP, where it's clearly present on the DC. And so that kind of got us a little confused because that's definitely the main thing we were thinking would happen. And so then we finally started to look at other cells, and we did a IL-5 driven CRE that targets Th2s and ILC2s. And there we started to see a drop in the IgE induction, which was very similar to if we took an ILC2 deficient mouse. And then finally, if we did a ROR alpha CRE, which is heavily present in expressed in ILC2s and Th2s, it kind of goes all the way back to the same as a full knockout. And so clearly, there's some IL-5 and ROR alpha positive cell that's important here. At affecting the IgE. And so we wanted to look, okay, what's a little bit more upstream of the IgE itself? And so we decided to kind of test these different mouse lines and their ability to kind of induce a T cell response. And so we looked specifically at TFH induction. And in all three of these, IL-5 driven or ROR alpha driven conditional deletion or the ILC2 knockout, we see a deplete or like a reduction of the TFH formation. That's happening in these mice. Um, and so we do think that basically there's likely some ILC2 deficiency or ILC2s have some sort of impact on the initial activation. Um, and so basically what we're thinking now is, right, IL-33 is coming in from the serum and at some level affecting the ILC2 population, which I didn't show this, but they basically expand heavily across the whole mouse in the secondary lymphoid organs. And we think that this basically is likely producing either IL-4/13 or perhaps some more surface-bound molecules like OX40 ligand that help kind of promote the activation of TFHs. But because of the fact that the ROR alpha CRE, really depleted it to the same level as the full knockout, made us wonder whether there's some other ROR alpha positive cell that's really helping to drive this. Whether it's an antigen presentation cell itself or not is still unclear, or if just the ROR alpha CRE is just more efficient at deleting than the other strategies we utilized. Um, but basically, the TFHs will then go into germinal center, and perhaps ILC2s may contribute more 4 and 13 to the germinal center, which can then also lead to the switching to IgE. And so the main takeaways, right, is basically that remote priming can exist, and that inflammation generated in one place can go elsewhere, and specifically there are certain cytokines that if released can actually induce an adaptive response. And when it relates to skin damage, it usually tends to be associated with allergy kind of realm of antibodies. It tends to be, require alarmins, which are both necessary and sufficient for this to happen. And likely ILC2s are involved to some extent. And then kind of moving forward, we kind of just investigated one way in terms of serum trafficking of cytokines. But we do think that there are other possibilities of the way, this might be working that definitely warrant some investigation. Namely, the trafficking of cells, whether it's ILC2s trafficking or dendritic cells or other APCs trafficking the antigen to other places, or seeing cytokine in one place and then going into a different tissue where they're seeing antigen. Or a kind of, for a lot of people, the more interesting aspect is what about the kind of neuronal transmission of, obviously a lot of these skin damages are associated with pain and itching and things like that. So could that basically get circuited and induce the activation of alarmins in other places, all the way down in the gut to basically activate the adaptive response. And then I think there's still kind of this, a little less satisfied with how it was fully identifying, what was that original antigen presentation cell. So there's still a little bit of work there to try to figure out, exactly who that is. And I think another question is, how are the ILC2s really positioning themselves either at the T cell activation interface or the B cell switching interface within the lymphoid organs? And then, how do other cytokines influence this remote priming, or kind of establishing resident immunity, are kind of different kind of questions that I think are interesting from where this goes. And then to finally kind of backtrack to the beginning here is when we think about type 1 and 3 responses, antimicrobial responses, they're really evolutionarily conserved. And these pattern recognition receptors are there to kind of try to identify the bug before it even causes a lot of damage, and to mount a response before it can get too worrisome. Whereas type 2 responses tend to be more of a, we don't know what's going to happen. So we can only figure out, when something happens, what do we do about it? So it really is there to detect disruption and try to restore homeostasis. And we do think that these damage-associated patterns can release a lot of these endocrine mediators to try to resolve that across the body. And then, of course, finally, just kind of, these two types of responses are often coinciding at the same time. So it'll be interesting to see how they end up regulating each other. And then with that, to not go too far, I'd like to thank Andrew, whose lab I was working with, and as well as Anna, who was the kind of dermatologist that kind of helped co-lead this project, and the rest of my lab. And then I'd be happy to take any questions. Yeah, thank you.